Our next speaker is Dr. Bill Andrews, who is a molecular biologist and gerontologist whose career has centered on searching for a cure for human aging. He is the founder and president of the biotechnology company Sierra Sciences. In the 1990s, he led the team at Geron Corporation that was the first to successfully identify the genes for the human enzyme telomerase. He's going to be speaking about the sources of aging. Here's Dr. Bill Andrews.
Hello, everybody. I've spent years talking about telomere biology at conferences, but today I've decided I would just like to talk about aging in general. I've been just in discussions about aging all my life. It's been a lifetime of discussions in dorm room chats at high school, college, graduate school, and I've kind of developed a lot of my own ideas about what aging is, and it's become an obsession of mine to pursue it. One thing that I have learned is that no one really knows a lot of stuff about aging. What is aging, why we age, what causes us to age, and how can we treat aging? These are things that a lot of us pretend sometimes to know, but I personally don't think anybody really knows this, and that's what I've been trying to figure out my entire life. I consider it a great adventure. I liken it to the search for the source of the Nile. When these explorers first started searching for the source of the Nile, and somebody asked, how long is it going to take to find the source of the Nile? They couldn't answer that question because there were too many forks and branches and dead ends along the way that they anticipated. And there was just no way they could predict that. And so the same thing is true with aging. Now, I and others, we're on a search for the source of aging. And now we can't say there's only one source, so I'm going to just change this to in search of the sources of aging. And I like the fact that we're all working together to solve all of our own aging, but first we have to understand it. And so through all my years of trying to figure this out, I want to go along and just discuss my best guesses as to what's going on. The first thing is, what is aging? Again, I don't really think anybody knows. A lot of people are inventing markers, but are the markers really associated with aging? Will reversing the markers reverse aging? Will reversing aging reverse the markers? I don't think any of us really know. But I got one question. It's not really a question. I don't think I need to tell anybody which photo of Betty White was taken first. I think everybody knows the photo on the left here was taken before the photo on the right. But I don't think anybody can really tell me how they know that. I mean, there's a lot of different things we can see that are different, but really, we don't know. I don't think any marker can be used to discuss what is the difference between these two photos. Well, I consider that aging, and I think that's the reversing of aging, the cure for aging. And so I call this the Betty White test. And as far as I'm concerned, nobody has figured out what aging is until somebody can make somebody pass the Betty White test. And so that's been an obsession of mine my entire life. The next question that I have is, why do we age? And this comes along with a lot of background of mine in evolution. My PhD is partly in molecular biology and partly in evolution. But I've been thinking about this, again, for a lifetime. And I'm just going to describe 4 bullet points here that kind of just give the logic behind what I think is the reason why we age. The first bullet point is, a successful species is one that can survive a rapidly changing environment. That's one of the bases of evolution. Number 2 is, the more a species shuffles its genes by breeding and mating, the more likely it is to survive a rapidly changing environment because of all the different combinations that can be created by breeding. Now, a third point is that more shuffling of the genes can occur if the offspring are allowed to interbreed among themselves more than the parents are allowed to rebreed. That's a fact, because if the parents rebreed, there's not very much shuffling, not nearly as much as if the offspring are allowed to interbreed. And the fourth one is, so there's a benefit to eliminating the old. A species that eliminates its old is going to have more gene shuffling. It is more likely to not become extinct just because it's got more variation in its gene pool, and it increases the likelihood that at least one member of its species will survive. And I believe that's why we age. So the next thing that I want to go to is what causes us to age. And there's a lot of causes of aging, which I'll come back to in a second. And they cause a lot of symptoms of aging. Now, a lot of the symptoms of aging are also causes. So there's a lot of causes, things that cause us to age. But the final outcomes of these are that we suffer a decline in health, a decline in appearance, and death. And that's what's going on in the aging process. Now, the causes of aging. Well, one is the environment. And I put here in parentheses oxidative stress and inflammation, but these are probably only some of the environmental impacts, because there's internal impacts, environmental impacts inside our cells, and also in external environmental impacts. Now, other things that cause aging are our genetics. We all have different genetics, and some of us have genetics that allow us to live longer or potential of living longer than others. So genetics plays a role too. And as most of you know, I strongly believe that telomere shortening is also a cause of aging, a major cause of aging. That's one of the more difficult things to control. Now, the symptoms of aging, you've all heard these many times, are cell senescence, mitochondrial dysfunction, altered gene expression, and many other things that you've heard in numerous talks if you've been attending conferences on aging a lot. So the common thing about all these things that I've just put in this red box is that all of them make our cells unhealthy. And so here comes a dilemma that I always foresee. And to explain this dilemma, I want to first think about bacteria. If you put bacteria in a petri dish to let them grow, and you mix these bacteria with unhealthy bacteria, after a certain amount of time of these cells growing, these unhealthy bacteria are going to grow slower than the healthy bacteria. After a certain amount of time, the healthy bacteria are going to overtake that culture. So there's going to be, even though there was 50-50 ratio to begin with, the ratio is now overwhelmingly in favor of the healthy bacteria. Then if we continue that culture even longer, the unhealthy bacteria completely disappear. So why doesn't this happen in humans? Why is it that we have all these things that happen? Our cells get unhealthy, our tissues get unhealthy, but we can't seem to fix those problems. We can when we're younger, but we can't seem to do it when we're older. And the reason is because human cells don't divide indefinitely. That was discovered in the early 1960s. I think I learned about it in the early 1970s. And I've been obsessed about that whole phenomena ever since. If we could figure out a way to make it so that our human cells would divide indefinitely, we should be able to overcome a lot of the things that I just described as the symptoms of aging that are also causes of aging. So I believe, and I think the literature, public literature strongly supports this now, that the reason why human cells cannot divide indefinitely is because of telomere shortening. And that is a major focus of Sierra Sciences, one of the main things that we work on, trying to solve that problem. Okay, so now let's go to the next question, as how can we treat aging? Well, there's a lot of things we can do, and Sierra Sciences is interested in all of them. One is we can minimize the impacts of the environment. We could regulate gene expression, figure out how to turn genes on and off to put them back to the way they were when we were young. We can use gene therapy to correct or supply missing genes that aren't working as well as they used to. We can correct cellular and tissue damage. We can do gene editing to make it so that we can get control over the expression of genes or inactivate gene variants that are not healthy for us. There's also stem cells. But one of the things I think about stem cells is it's really just a way of providing our body with cells that have long telomeres. And therefore, we come to my final one, which is telomere maintenance. So again, Sierra Sciences is focused mostly on the bottom one, telomere maintenance. And if you don't know what that's about, you can listen to a lot of my presentations on my website, which is shown there. But there's also one thing that you'll hear a lot is that people say that by lengthening telomeres using the enzyme telomerase, you can cause cancer. I don't have time to discuss that today, but you can go to my book. So telomerase and telomere lengthening I believe, is more likely to prevent and treat cancer than to cause cancer. And if you want to learn more about that, you can go read my book, especially pages 64 to 77, called Telomere Lengthening. Or you can watch a presentation where beginning at 25 minutes and 25 seconds, I spent about 45 minutes just discussing in excruciating detail why lengthening telomeres actually is more likely to prevent and treat cancer than to cause cancer. So on our quest to find the source of aging, how are we going to know when we get there? Well, we actually have no good way to measure the slowing of aging. A lot of people are working on markers. I think these markers are very important. I don't want to sound like they're bad things, but I think they're going to tell us more about our health than they are about our aging. And so I think we need to do more, something like the Betty White test. And I do strongly believe to know that we are really treating aging and that we really understand aging, we have to show that we can reverse it. And when we do that, then we will truly know that our search for the sources of aging is complete. Thank you very much.
Thanks, Bill.